today I'm going to continue with biology of the lymphocyte, B lymphocyte. So we kind of building up a story. Now, in today's lecture, we will cover development and differentiation of B lymphocytes, where they are synthesized, and we'll go a little bit in detail. And then what are the molecules expressed on the surface of B cells. And then again, the most important thing is B cell signaling, because that's where we would want to use the drugs. So signaling is a little bit complicated, and I keep on telling you in physio as well that it's going to be like a transition. So help yourself and try to memorize cram, whatever you want to do. I'm available. As I said earlier, I'll help you out. I do carry quite a few notes and whatnot to help you learn signaling. But this is something that you need to know as pharmacists to understand where the drugs are acting. So drugs are not going to act on B cell. They're going to act on very, very specified proteins. Okay. Now you're familiar with a B cell. And when I say B cell, I'm pretty much talking about a cell which is lymphocyte. A cell that's something to do with antibodies, production of antibody. Immediately, that should stick in your mind that B cell is something to do with antibodies. Now, also remember that this particular cell, which is geared for antibody production, will synthesize an antibody in response to an antigen. We discussed that yesterday, the capability of diversity or specificity that has. And that again goes back to those particular variable portion, whether they be on heavy chain or light chain. And then also remember that there is I keep on telling you, like a computer tag, those kind of arrangements there, where everything is unique. Everything is unique. And that's very specific, tailored for a particular cause. And why do we need to know that? Because we want to make vaccine. We want to make very specific vaccine. We don't want to give a very non-specific vaccine. So when we give you MMR or a flu, so on and so forth, we really mean it. So that's geared towards specificity. That's why things last for a long time. And we did discuss primary response, and we talked about secondary response and what are the antibodies produced, and why do we need to have a booster doses, so on and so forth. So in technical terms, when we give you MMR or a measles vaccine, we pretty much guarantee. So that's the only success story in the medicine is that we give you vaccine and you're totally protected. We have eliminated some of the diseases, like for example, smallpox. We are still struggling with tuberculosis, malaria, AIDS, so on and so forth. So most of the time, what you will come across in medicine and treatment of medicine is management. Diabetes, we're still struggling. There's no vaccine for that will kind of protect you. And then hypertension, renal failure, you name the disease, it's more or less damage control, and more or less management. But in this case, vaccination is something, when we give you polio vaccine, we eradicated for most part polio. So that is something that we need to understand. So the first line of treatment, or I would say prevention, when you say prevention is better than cure. So prevention is vaccine level. So that's where immunology comes. So that's a success story in this case. Now, the developmental part, I think by now I want you to get familiar with the schema that we've been talking about, all the cells. And you will see that over and over again. And it's important for you to understand the concept behind that. If I was to tell you it's a T cell, pretty much the same scenario, lymphocyte, you name a cell, cells don't pop up out of the blue. They go from a series of events That takes time, right? And then there are stages for differentiation. So right from primary cell to the last cell, there are differentiation. And I'll give you an example. Is that, for example, in cancer cell, uh, they are not regulated, and you may see cells which we'll call undifferentiated. This means not fit enough to do the purpose, because by the end of the day, we want a mature cell, a cell which is capable to function. But bodies don't produce mature cell. Bodies will take, it's like developmental stages, intrauterine life, even the babies, 9 months. It's a stage process. You want to deal with it individually. Now, in immunology, when we draw a cell out, and you want to make sure what stage is it at, we go for a procedure which we say expression of membrane immunoglobulin expressions. We look at the membrane. We could kind of look for the membrane, and we have a test. We will talk about that, and I want you to read ahead of me. I'll post all the information. That we take your cells out and we look for specific proteins, right? Okay. So this is what we do as a testing thing. And the other important thing is you have to know that right from the stem cell till a mature B cell, we already discussed about the VDJ gene rearrangement. As I said yesterday, there are also steps, right? So all these steps have been followed and cells been matured. So two series of events happening at the same time. Now you can see two important, I would say three important stages for B cell, right from the stem cell, pro-B cell, pre-B cell, immature B cell, before you get the final B cell. And all these cells are in your body, right? And if your regulatory system is not working and you don't have a control, you may get a cancer where you get an immature or a pre-cell or a undifferentiated cell in your body which would not perform as it ought to perform. Now, so you can see from here, when we talk of pro-B cell, we're talking about the first junction. Remember I said D and J are the first, they pair up. D and J, so that's pairing taking place at pro-B cell. And then V pairs up with this pair, right? And we end up getting VDJ. So if I was to look at this cell and it just paired up a DG segment, which is a diversity and joining segment for heavy chain, because we're talking about heavy chain in this case. You can see all H, right? So heavy chains are made in pro and B cell, and you can see from here, over here as well, that when we are making this, because remember these BCR is an antibody. We already discussed that. So we look at the membrane protein outside the B cell, which is an antibody, and we look at this antibody and we say, hey, you know, just pay attention to this one. Don't pay attention to this double bar, because it's something that I'm going to discuss today. Okay, but this one over here, as antibody, and I also told you that there are two antibodies which are, I'm going to use this word today, constitutively, by constitution of the body, that these two antibodies will be expressed on B cell, IgM and IgD. Okay. Now the first one to appear is IgM, as you can see from here, but the heavy chains are there, which will determine as to it is micro. We already know because micro is a heavy chain. And then when we get the light chain coming over here, then you have a complete IgM. Complete IgM. But remember, if you have only expression of, I'm going to ask you this question, if I have a cell, which is a B cell, and I only see IgM there, and I do not see an IgD there, so what is the difference? The difference between a cell having IgM is still immature. So you have to have both expressed on the B cell to be called mature. So a mature B cell will have both. And you can see from here. Okay, then again, just remember for now this double bar, I'm going to come to it in a while. And then again, you have IgM with all the chains there and IgD, and we call this cell a mature cell. Now, there are different types of cells. Let's divide B cell into two cells at this moment. One is a memory cell and what is a plasma cell. Memory cells are those cells that are mature cells. And again, I'll come back to, I think I must have told you at the end of the new book, there's a glossary for definitions. Make sure you read it up because you need to understand the concept. And many of the questions for the first part of immunology are going to address your basic understanding of the concept, right? So when I say memory B cell, so the difference between a memory B cell and a mature cell is that memory B cell has seen an antigen. So it keeps the record of that antigen in the memory. That's the memory. So once this B cell is going to see that antigen again, it's going to blow up. This memory cell will become a plasma cell, swell. It's like you give an antigen the primary response. One of the things that happens in the primary response is the body makes a memory cell. Before a primary response, there's no memory cell. When you get a secondary response, memory cell is already there. So when you expose the body to another antigen, it takes out the file and B cell sees it already there, it blows up. It becomes plasma cell and starts producing antibodies. Depending upon the need, class switch, it is designed to make IgM and IgM and D, but again, it can produce any antibody as needed. Okay, that makes sense? Now, if I was to look at the test, which is called fluorescence activated cell sorting, it's called FACS. We have this on campus, we don't have it over here, it's in biology. Fluorescent activated cell sorter. We take a B cell and tag it with a fluorescence dye and then see under the microscope or machine, it picks up. So it kind of shines. So in this case, what we see in the membrane expression is that if I have an antibody, so I'm going to take a cell and put an antibody on top of that. Or a fluorescent, let me put it like a fluorescent label on top of that. Now this fluorescence label, which is going to shine, is only specific for IgM. So when I take that and put over here IgM, it shines. So I know that it is an immature B cell. If I was to take two labels, because I cannot use the same label because they are very specific. So I have two labels, one for IgM, one for IgD, put it on my cell, and guess what? Both will go because they express the receptor, IgM and Ig, so I will conclude that it's a mature one. Okay? And then again, if I was to look for any other particular cell, like G, A, M, D, E, I have to use that particular fluorescent tag that is designed for that antibody. I cannot kind of mix and match because these antibodies are very, very specific. Make sense? So that is the basis of the test that we will do that. We'll discuss that. Now, let's say if I take a cell and put an antibody I put all these antibodies there, and no antibody gets tagged onto the cell. So guess what? What is the cell type if I don't get any antibody labeling? Stem cell. Stem cells very difficult to have it in the labs. Correct. It could be either a pro or pre because they don't have fully developed antibodies. So that's what the basis of the test is. Does that make sense? Okay. If I was to take T cells and put these antibodies on top of those T cells, would I see something? No, because they are B cell related. Though, technically, there's a term I'm going to use for a test today. is called false positive. You do a pregnancy test, it becomes positive. It cannot be positive. It wasn't a planned pregnancy or there was no intercourse, whatever. But so this means you say, hey, I know I'm not pregnant, but why this test is giving me positive? So that kind of problems that we have to test in is called false positive. On the other hand, a person has a large belly and you sure this baby is moving in the uterus of the mother, and you test the pregnancy test is negative. It cannot be negative. So that's a false negative. So the things can. So likewise, I took an example for pregnancy. It could be for hepatitis. You may get this false positive and false negatives in the testing system because of, if I was to ask you, based upon what I've taught you so far, concept question, where is this false positive and false negative coming from? If I was to ask me, explain one mechanism, one immunological mechanism that's responsible for this false positive or false negative. I talked about specificity, that's still there. I talked about diversity, that's still there. But something else I talked about, yeah. I also talked cross-reactivity, where an antibody may identify a wrong antigen, possible. So cross-reactivity, that's where you want to make sure, right, that is there. Okay, so far so good. Now we don't leave things over here. I'm going to introduce, and I did introduce, a very specific, as I said, I think I did talk about that, the computer tag or the bar over there, which they call computer tag. In immunology, we call CD. So every cell of our body has a CD, and as you look at your computer tags over there, so you will see a series of bars. Okay? Just by looking at it, they all look alike. But if there are 1 million items in Walmart, they're going to come up separately just by tagging it within that 1 inch or 2 inch of the tag, the bar. Now, these cells have that, and these are proteins being clustered together. I'm going to use the word cluster, right? As you can see, I just show you 2 bars over here, right? So there are tons of bars over there. When we scan them, and we have a number for each and every bar. It is called CD, cluster of differentiation. We call CD. And that's where, if you have heard about CD4 T cells, any number that we give. Now, I think you may want to sharpen your memory skills or develop memory B cells to remember quite a few of them because they are important and drug targets, right? So I'm going to say CD and I'm going to give you a number. I don't expect you to understand millions of them, but I would expect you to know the one that we use as drugs. Correct? Now, let's exercise. So 1, 2, 3, 4. 4 important CDs, right? Which are expressed, CD molecules which are expressed on the surface, CD10, CD19, CD20, and CD27. Now, if I was to tell you, for example, that you can see from here, till this stage over here, till over here, we have CD10 expression. Then we have all the way CD19 expression. Then we have only CD27 expression on the plasma cell. So we have another CD differentiation, not based upon an antibody, but also based upon the computer tag. You're going to tag it, it's going to say CD19, 20, it's going to give the price, right? So 19, 10, 19, 20, 27, and let's see what comes up. Okay, now, I mean, honestly speaking, I cannot come up with a better way of teaching immunology because I may have something in my mind, but I would rather use the book as the standard. So because it's going to be easy for you to read. So book, but unfortunately, because I review textbooks as well, it goes back and forth, back and forth. it's like following basketball game. It's like a basketball game, but that's the way it is. So you may want to get used to it or read before the lecture or after the lecture. So development of B lymphocytes. They're called B lymphocytes because the first time we found what it was discovered in the birds in a bursa which is called fabricius. Bursa fabricius, that's what we call it, B cell. Bursa derived, but now we also call them B cell because we think maybe they are produced in bone marrow, but that's not the correct origin of the word. And in the previous figure, we discussed about the developmental checkpoints, meaning is that there can be arrest at any stage. There can be an arrest or problem at any stage. It's like assembly line of the product of car or whatever. So you're going to go back and say something went wrong over there. But the early B cell differentiation in human happens in fetal liver before birth, and after birth, it is a primary lymphoid organ, which is bone marrow. Bone marrow is so these are in terms of where those B cells are situated. Okay, now I already, just like a recap, remember I said that some of the slides that follow the picture or figure are just a recap of the figure. I may have already explained that to you. If I have missed something, this is just for me to kind of allude to those ones. So we already discussed about pro B cell, and we talked about the chain. We talked about two markers. CD19, if you go back and look at it, it's expressed on all stages except plasma, because plasma had CD27. If you see that bar, it will show you. When you look at a pre-B cell, again, you will see that they all have a heavy chain, VDJ, and there are two markers, 19 and CD19 and 10 expressed on them. So we will differentiate between pre-B cell and pro-B cell based upon, well, you can see right now it doesn't really explain much, but you will see over here as we move along some other markers in terms of cluster differentiation, because pre and pro are too close to differentiate on the basis of CD10, 19 marker. Okay, so we may kind of mess them up in terms of identification, but probably we don't need to go in that detail. Now, yesterday when I talked about, and this is normal protein transcription, we talked about VDJ recombination. That's what we discussed, right? And also told you that they are regulated, controlled. But the thing that you have, I'm not going to explain that to you in this first part. I'll explain that to you in clinical immunology. What you can ask yourself when I explain to you normal, you can ask, what if? What if it goes on? What if? What if? So it's a what if for everything. That what if is a pathophysiology. But we are not prepared, we may not have time, but you should ask this question, hey, what about the VDJ rearrangement if it doesn't go right? So there's a homeostatic mechanism in the body, right? So one of the processes that we taught you in pathophysiology is that when things go wrong in the body and things are not properly arranged the way the body wants it to be, then we have a system in the body which is called programmed cell death. That is the removal of unwanted cells without causing what? Huh? Louder. What did you say? Okay. Use a better term than harm. You're teaching immunology. No, we cause them death. I want to hear a word. Huh? Cancer. Without causing inflammation, that is the major difference between necrosis and apoptosis. When you remove cells with necrosis, you get inflammation. When you remove cell through apoptosis, there's no inflammation. If these things would have happening, we would have fever all the way through. We will all be sick. Body doesn't want you to give you a fever, because then there's a fever, there's going to be clinical signs for inflammation. So it's a very neat way of killing. All your red cell get killed within 4 months and replaced by new cells. You get fever? Do not. Do you know that? You don't know that. So that's happening with regeneration with programmed cell death. Okay? In immunological term, we use the term, if this rearrangement is productive, if whatever match we do, hey, goes well, we will let it stay. If it's non-productive, and if that combination is not the one that is going to give you IgM or IgD, then the cell will kill it by a process of programmed, because that is programmed in the body, that's fed. It's just like a computer program, it's programmed for that. So body has a special program we call apoptosis, and it's called programmed cell death when that thing doesn't go in a way they want it to go. Okay, now we already discussed that. I'm going to pay attention to that bar that I showed you in the first picture. Okay, now I want you to visualize there's a B cell, there's a receptor over there, the antigen comes. Now what happened? That's what we're going to discuss today. What happens when a B cell sees an antigen? So the B cell will see an antigen through a antigen binding site, which is this one, right? And something should happen that will convey a signal to the nucleus, because by the end of the day, all those things that need to happen in terms of a response happen through the nucleus, because they're all nucleated cells. Okay? Now, antigen binds over here, heavy chain, light chain, we already discussed that. Now, once the antigen binds over here, there are two parallel bars that they sit next to the BCR, right? And I usually kind of give the example for you to remember. If this is a pilot, so this is a co-pilot. It's just like a plane. I don't think any of the airlines will fly a pilot without a co-pilot. That's actually their rule. Okay, but remember the co-pilot has the same license as a pilot, so they both are pilot, but one is called co-pilot. That's the way it is. And where does the co-pilot sit? Left or right? Right. That's the rule. So most of the time, I mean, I'm just giving you some mnemonic, the co-pilots are going to be on the right-hand side. And we'll explain that how the sequence of signaling work. But right now, it again is a polypeptide chain, as you can see from here, disulfide bonds, and it is a part of the immunoglobulin superfamily, Ig. That's why it looks familiar. Two legs. It's two-legged. It's not like a four-legged animal, it's two legs, so kind of human, so to speak. Now this portion is called Ig alpha and Ig beta. Okay, and there's everything that I give you may have another CD number. Sometime I'm gonna ignore that, and sometime I'm gonna tell you, right? You cannot make it up if you're gonna read it on your own. So make a note of it. It's not given over here, but based upon the drugs, as I said, I'm gonna let you know if this is important for you to know, right? I don't want you to know CD79 A and B, but I want you to know the other name of Ig alpha and beta. So this will get triggered and it will send in signal to the nucleus of what needs to be done when you see an antigen over here. This is called signaling pathway, signaling transduction, or outside in signaling. When an outside signal triggers the response, we call it outside in signaling. Okay? So if you go back on the pre-B cell receptor again, you can see, now you can see those two bars, because I didn't talk about that at that time. So they have Ig alpha and Ig beta. You saw that over there. So these are called signal transduction molecules because they do a job which is signaling, signal transduction. So anything that comes, it's just like I'm going to give this to him and say, pass it on to her, and him and her. So it's going to pass through a sequence, right? A what do you call ballet. So that kind of a signal transduction molecules, there's a chain of events. So it says transfer signal to the nucleus, but it's not that simple. It's going to be very complicated, and we're going to discuss that, right? And as a rule, do you wish it was simple? Well, you would wish it's simple because then I'm just ask you one simple question. But the problem will be then you're gonna be out of job as a pharmacist. You wish it was complicated, so it creates more jobs for you. More drugs, more jobs, more pharmacists, things of that nature. So think of that in a positive way. But I can assure you is complicated. Okay? But right now it seems to you transmit signal, right? And if this signaling pathway, again, same thing. I don't need to ask you every time, what if? What if it fails? What if? So what if, I've got two answers for you. What if is that if this thing doesn't happen, right? We have a disease which is called Bruton's tyrosine kinase. Problem with the Bruton tyrosine kinase. And remember these signaling events that happen, there are enzymes involved. I would rather want you to remember signaling enzyme than I want you to remember enzyme for electron chain transport, for example. Okay, because that's like a theoretical thing, but this is like a pharmaceutical thing, which is in more interest for you. Okay, so enzymes are there. So enzymes are protein. If you miss, there's enzyme, right? For example, there's one disease we'll discuss one full lecture on genetic disorders. Okay, so one thing is X-linked A gamma globulinemia. Without telling you, just by looking at the word, and you guys are doing the medical terminology as well, right, Dr. Hussain? Right? A gamma globulinemia, what does it mean? The gamma globulin is in the blood, but it's A, right? Is not normal, either absent or deficient or defective. And since it's an X-linked, go for the male-female, XY-XX genetics. So this will show you, I don't need to ask you that this particular disease, would it be transmitted? If transmitted, it's going to go to the boys or the girls based upon the who carries that X gene. So that's what the genetic disorders are like. Okay, now the other important thing in the same picture that I show you, when an antigen sees an antibody, right? Because I think when you saw that figure of production of B cell, probably you thought that these are the normal B cells which are made, which is true. But also keep in mind the same differentiation step go when an antigen sees an antibody and you make excessive IgM or IgG or IgD or IgE. So the same process goes. So antigen sees an antibody, right? And then there is recombinase. So remember that enzyme we talked about, recombinases. That's going to make those choices. And then there's another term I'm going to use today, which I didn't use. It's going to pick up. It's just like I'm going to write a word, so I'm going to edit words that make sense. So this is, we call it receptor editing, because that portion for the Fab fragment for antigen binding has a receptor there, so that needs editing. This means I have to gonna give you a number there. So that's what the system does, especially for enzymes. Now, again, what if? What if things go wrong? What if it generates a receptor? Was a part of mnemonic something that I need to know? Or are you recording my voice? Generation of a receptor reactive to self-antigens. Now what happens, you see an antigen, hey, this is antigen XYZ. Okay, make an antibody for XYZ. You're going to kill this antigen. Then the body says, wait a minute. The same antigen is expressed on cornea of your eyes. So do you mean that you're going to kill that virus, antigen, and you're going to kill your cornea as well? So no, wait, we don't want to do that. We'd rather have that bacterial virus than to lose the cornea. Understand the point? So that's where it happens. If it by mistake generates that kind of self-antigen, we delete that B cell. The system says, stop it. Garbage it. We don't want this B cell to continue because it's going to mess up the whole thinking. Right? So we don't want to generate any reaction to self. Because if it does, we need to kick it out from the bone marrow. But again, it may happen when you may have a genetic disease. Okay? Yes? Now, I'm going to use another term today, and it's going to be bombarded with different terms and definitions. You must have heard of energy with E, but it's energy, A with energy, right? So keep in mind, it's not don't come in the exam and tell me there's a typo there. It's not a typo, it's the immunological exam. Energy. So when an interaction of an immature B cell with a soluble self-antigen, right? So many a time that your B cell sees an antigen which is circulating around, and it happens to be a self-antigen. So it's going to ignore that. Otherwise, if it doesn't ignore that, it responds to all the calls that they normally get, right? It's like my little child's going to call 911 and they're going to respond to it. Do you think they respond to every 911 call made from the house? No. They really want to verify who is it calling. Otherwise, it's going to be a joke. So body doesn't want to react to everything. It's going to make sure. But what is the process called? This is the process which is called energy, which means long-term inactivation. We don't want to respond. So even if you're going to use one number and call 911 over and over again, it's going to put your number in a way that, hey, be careful with this particular number, right? Okay, so body does it. So this process of long-term inactivation is called energy. Okay, now let's see picture thing again. I'm going to go back and forth with picture plus editing. Now, this is a immature B cell which has a receptor, BCR, right? It sees a bacteria comes in. So bacteria is like a cell or any cell. So it has all those antigens. So they're all antigens around bacteria. We'll discuss these antigens in detail. That's why even in bacteriology, virology, you need to know immunology. You're going to be tested for the first part, but again, every pathophysiology needs immunology. So you need to know, I'm not going to teach you right now what are the antigens present in Staphylococcus aureus or Escherichia coli or so on and so forth. Just a generic thing that they're antigen, they are interacted with the B cell receptor, and then two things can happen. One thing can happen is that if the body finds that this antigen is similar to what we have as a self-antigen, so this will stop this B cell. Hey, don't process this information. Because if you do process this information, you're going to cause damage to your own tissue. So that's we call delete it. So we delete those files. That's the term we use, deletion, in immunology as well. We delete that information. And you can see so what we mean is we remove those cells that may be self-reactive. Now I'm going to throw a very difficult concept for those of you who are with me or who are not kind of somewhere else. What happens is that if there is a chance that the B cell can generate some of the antibodies which may recognize self-antigen and the system fails to delete it, there are ample chances for you to get autoimmune diseases after infections. One of the causes of autoimmune disease is genetic, fair enough, but you may get autoimmune diseases by infections because those foreign antigens can bring a change in you, and that's very common that we normally see for some of the diseases which we'll discuss later. Okay, but nevertheless, they are deleted. Or if the body wants to keep it, it's going to keep it. Otherwise, want to make sure that this does not respond, because if it starts multiplying, especially for the self-reactive antigen, and they are not deleted by apoptosis, for example, let's say the other way. What if? Because it's not a perfect system. It's the best system though. So this doesn't get deleted, and you still get a BCR specific for self-antigen. So this is not in the figure. So this particular cell which is not deleted can stay and hide somewhere and can be recalled upon later in your life. So you're normal, everything is normal. All of a sudden, you are 20 years of age, young female, and she develops multiple sclerosis, which is an autoimmune disease. Why she did not have it in the first place? She had genetics for that. She had ability, but cells were hiding, and all of a sudden they decide to, hey, let's come up now. Okay? Now this picture tells you if it is a BCR, which is not self-antigen, and it is an antigen for that particular bacteria, right? It can now leave bone marrow and divide. So there is that system over here that tells the body what to do. Because remember, if you start making antibodies to every antigen, so all of your body is an antigen. So you're going to carry loads of antibodies, which happens if this is not normal. Okay, now let's say the question you have to ask yourself is, where do they meet? Where this antigen antibody meet? So you can see from here, most of the time when we say that where do they meet is a lymph node. So we have lymphatics, antigens picked up from everybody, carried by cells, and put it into a lymph node. So lymph nodes are scattered all over body, and you can see an antigen comes, and there is a B cell captures antigen over here, and then it starts processing that. Okay. So the B cell seals and starts processing it. But also remember, in an antigen, in a lymph node, if you know this one slide, I think, there's a B cell area, there's a T cell area. Normally they operate separately, but just like mom and dad, they're going to be there in the family. They want some of the decisions to be made as a family decision. So in this case, and there's a good reason for B cell to ask help from T cell. What's a good reason for the B cell to ask help from T cell? Sorry? Cytotoxic. Okay. Yeah. So B cell recognizes antigen. That's not a problem. No. The question is, why do the B cell need to consult T cell? Yeah. Yeah, but why? Very good. Because T cell only has the genetic information, not the B cell. Because T cell has MHC. Very good. Okay. So in this case, the T cell may want to say, hey, you captured an antigen. Oh, wait a minute. Let me see it as well. So both of them dock onto that antigen and see it from both sides. And then they respond if the need be. So there is another term I'm going to introduce today, which is that the B cell talk to the T cell. But on the other hand, I also told you there's antigen presenting cells, which are professional antigen presenting cells, like dendritic cell. So they will capture antigen from nowhere and bring it to the cells. One of the cells they bring it to is a T cell. Because as I said, T cell would not see a soluble antigen or any antigen. Antigens are food for them. They just want a protein. So this means protein cannot be presented. Protein can only be presented if there's an MSC. MSC is only with the T cell, and sometimes you have to act together. So you can see this, a triad where a B cell in a family can respond, and it does respond, but many times it wants to consult a T cell, but T cell has other player over here, which is an antigen presenting cell. All are sitting in the tissues. So there is an interaction taking place. Right. Now, another term I'm going to use right now is that a mature B cell or a mature T cell, I remember I used the term other day, naive or virgin. Any cell which has not seen an antigen, so it doesn't know, it's just naive, it's immature. So once he sees the antigen, it gets activated. So we call that cell activated B cell or T cell. Okay, so you can see that once these cells get activated, they're going to go and respond. So this response we call immune reaction. immune response, same words, or a better term, effector response. Effector response. So all the way from antigen to the effector response. Okay. So what I've told so far, they're antigen-specific B and T cells, right? I said those have seen that antigen. But also remember when I showed you earlier that all your body antigens are introduced to the T cell and given them this data. Make sure that you don't react to the cell because they are self. The concept of self versus non-self, right? So they pretty much have the information. They should not react. But when we say antigens, we use another term. If the antigens are proteins, they could be anything. They are known as thymus-dependent antigen. It's the same way of saying that another is same thing said in a different way. Okay? An antigen. So antigens are thymus-derived because that's what the T cell. So any antigen which is a protein. So now I'm going to go back. I just said earlier, why do B cell need T cell help? Well, B cell need T cell help, especially if they're going to deal with proteins. Because protein need MSC, they don't carry an MSC. That's what she told me. Good. Now, the T cells are made in thymus, which is called thymus-dependent, right? But let's say if the antigen the B cell has just picked up is not a protein. It happens to be a carbohydrate. Like, for example, some of the bacteria capsule is a polysaccharide, it's a capsule. Then B cell doesn't need T cell help. It's going to make antibodies. So we call that thymus-independent. So this is the same thing said in a different way. It kind of piggybacks to what I've already told you, that the T cell deal with proteins and thymus and so on and so forth. So we already talked about thymus-dependent sites where the antibodies are synthesized in the germinal center. And then again, this is what we already discussed yesterday about how the diversity and so on and so that happens. Now, if you look at this cell, for example, a lymph node, a germinal center, the lymph node, which has a mantle, outer covering, and this is a dark zone and the light zone. So one of the inflammation, your lymph nodes swell. So you cut them up and you will see over here that there's a proliferation. All those things that I've just told you, the antigens are coming. So it's like a police station. You bring people, do this. Many things are happening. So that's like our lymph nodes are different satellite stations, the police station. They want to make sure that nobody comes in, in terms of, having not the right identity. All these processes are taking place, antigen processing, T cell, all these things are happening where B and T cells are, it's like a family here, where they are responding. And if they do respond, especially if we're talking B cell, the response with all different things will be, hey, let's make a memory cell. So we put that data for that particular antigen in store. Let's make a memory cell. Or let's make a plasma cell, as I said, like a pregnant B cell is going to produce antibodies. And that's what we call it, cartwheel. You see like swollen cartwheel, the big belly. A B cell with a big belly over here is because there's a rich endoplasmic reticulum. So that's basically the reason. So we already discussed that. Last slide, I'm just going to take one more minute. Same thing is happening in all parts of your body. I just told you they're happening in lymph node, but they're also happening in your mucosal tissue. And when I say mucosal tissue, what antibody I'm talking about? IgA. IgA. So you can see from here, again, there are a lot of bacteria in the gut, and there's an M cell, there's an dendritic cell. It sees this antigen, it sends a message to both B and T cell, right? And then they respond. So if B cell have seen this antigen before, they're going to start making an antibodies, and then these antibodies will kind of come. If they want to have a IgA antibody, an IgA antibody will be made, and we already discussed how the IgA antibody is carried from the plasma to the lumen of the intestine. So whole processes are happening because in the gut, you may have viruses, for example. You may have bacteria. You have many things And then there are special cells like antigen presenting cells which will present it, and all these things are happening, if you know the gut histology, in the lamina propria or Peyer's patches, where all these responses are taking place. And also keep in mind, the gut basically is one of the major areas that sees the antigen, is a major area that has the maximum number of cells. Okay, so I will stop here